Alright, so we left off talking about true breeding varieties. So remember, those are going to be the plants that we know that when they are self-fertilized, they will create offspring that are identical to the parent. So now we need to talk about some terminology that I'm going to use a lot. So I made a slide for it individually. That way, if you need to refer back to it to see what's going on, you can do that. So hybrids in genetics are going to be the offspring of two different varieties. So if you had the purple flower mating with the white flower, that would be an example of a hybrid. Or if you had the tall plant mating with the short plant, that would be an example of a hybrid. Hybridization is the process that we use to produce the hybrids, and we call that cross-fertilization. So when we basically take the plant and cross it with another plant, not itself, that's called cross-fertilization. For ease of rolling off the tongue, we're going to call hybridization a cross instead of hybridization. If I'm talking about self-fertilization, I'll say that. If I'm talking about hybridization, where we're taking two different plants and crossing them together, I'm going to call them a cross. The P generation are the true breeding parental plants, so the P is for parental, so that will be the true breeding varieties, the ones that we know that when we self-fertilize them, they create offspring that are identical to themselves. The F1 generation, the F is for filial, which means son. So we always talk about daughter things in biology. In genetics, we talk about son things. So filial 1, F1 generation, that is going to be the hybrid offspring of the P generation. So it goes from P, parent, to F1, offspring. Okay, it's the first generation of offspring of the P generation. And then the F2 generation is the offspring that are going to arise when the F1 generation is either allowed to self-fertilize or cross-fertilize. We get the F2 generation. Another way to think of it kinda would be like your grandparents, your parents, and then you. Okay, so what were the 7 characters and their traits that Mendel studied? He studied flower color, flower position, seed color, seed shape, pod color, and stem length. I'm not going to sit here and tell you what all of these things are. I think you're smart enough to figure it out. You may not know what that flower position is, whether they are axial or terminal. That just means, are the flowers at the top of the plant, which would be terminal, like at the very end top of the plant, or are they axial? Do they kind of grow on the sides of the stem? So that's what the flower position is. But I think you can kind of figure out, the rest of it. Okay. So what was his experimental design? He performed a series of what we call monohybrid crosses. And from there he came up with some hypotheses. And we'll talk about those hypotheses in a little while. But a monohybrid cross is going to occur when you cross plants that only differ in one character. That's what the mono part of monohybrid is. So for example, he would cross purple flowers and white flowers together, but those other 6 characters on this slide were the same. So the flower position was either axial or terminal on the 2 plants he crossed. The seed color was either yellow or green on the ones that he crossed. The seed shape was either round or wrinkled, but not a mixture. He only wanted to have the hybrid for one of the characters at a time. That's the monohybrid. To be clear, when he crossed purple flowers and white flowers, he did not get light purple or lavender flowers, which shows that the blending hypothesis cannot be correct. Because if the blending hypothesis were correct, then every time he breeded purple flowers and white flowers, he should get lavender colored offspring. But what you'll see in a little bit is that he did not get lavender colored offspring. So the parent generation of purple flowers by white flowers, the first generation were all purple. However, when he took the F1 generation purple flowers and he crossed them together, He ended up with an F2 generation that had a ratio of 1 to 3 white to purple flowers. So 3 of the flowers ended up being purple out of every 4, and 1 out of every 4 ended up being white. And I'll show you a picture either on the next slide or in a couple slides of this test cross so that you can see what's going on. And so he reasoned that the white trait isn't just disappearing in the F1 generation, it's just being covered up or masked by the purple flower trait being present. So this is the dominant and recessive thing that I was talking about earlier, which I will fully present to you in a little bit. But he also reasoned that the F1 plants must be carrying 2 factors for flower color, because otherwise, if he wasn't, then they would either all be purple, or they'd all be white, or the F2 generation, so like the F1 would either be all purple or all white if there weren't 2 factors. But also, if there weren't 2 factors, then the F2 generation could not have shown whatever wasn't in the F1. So if the F1 was all purple, then, which it was, the F2 could not have possibly shown white. Or if the F1 was all white flowers, then the F2 couldn't have shown purple. But there's 2 factors that are being passed on, and only one of them is being shown while the other one is being masked. We call that complete dominance, where you have 2 different versions of the gene present, the alleles that we talked about with meiosis, but you're only seeing one of them. That's dominant versus recessive. That's complete dominance. There are other types of dominance that we'll talk about, but complete dominance is the first and easiest one that we will talk about. So here's the picture. So the P generation was a cross between purple true breeding flowers and white true breeding flowers. When you crossed them, all of the F1 plants ended up purple, but then if you fertilized the F1 generation against each other or to themselves, you ended up with an F2 generation that had 3 out of every 4 plants being purple and 1 out of every 4 plants being white. Now please understand that Mendel did not do this with 4 plants. He did this with hundreds and hundreds and hundreds and hundreds of plants. He had a huge garden. All the monks at the monastery helped him tend his garden, okay? Like, this was a lot of plants. I'm just showing you what happens out of every 4 because that's the statistics of it. If I tried to show you 400 million plants, you would be so confused. So please understand that he did not just prove all of this stuff by using 4 plants. It was many, 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 many flowers. Okay, so long story short, he did monohybrid crosses 7 times. He did it for all the different characters, and he was able to come up with 4 hypotheses from there. The first one is that there are alternative versions of genes that are going to account for all the variations in those inherited characters that are going to account for the traits. Those alternate versions of genes are called alleles. You already know this. And so homologous pairs are going to be made up of the same genes, but they are going to have different alleles. Sometimes the 2 alleles end up being the same. For example, if you have blue eyes, that means one of your parents gave you the blue eye trait, and your other parent also gave you the blue eye trait. That's all you have, so that's all you can show. But it has the potential to be different, or a different version. So those are different, you could potentially have different alleles. Like if your mom has blue eyes, she can only give you blue, but if your dad has brown eyes, he could give you brown, most likely, or he could give you blue, potentially. But if he gives you the brown trait, then the brown's going to win out over the blue eyes every single time, because brown is dominant over blue. Again, we'll talk about that in a little while, but I know you guys kind of already know those things, which is why I'm mentioning them now. His second hypothesis was that for each character, an organism will inherit 2 alleles, one from each parent, and like I was just saying, they can be the same or they can be different. If an organism inherits alleles that are identical for a gene, that's going to make the organism a homozygote or homozygous. Remember, homo means same. So they'll either have like 2 capital letters, so 2 dominant traits, or they'll have 2 lowercase letters or recessive traits if they're a homozygous individual. So that's how we'll represent them, and I'll explain that a little bit more in a second. Alleles that are different for a gene are going to make that organism a heterozygote or heterozygous. So they'll have one of the dominant alleles and they'll have one of the recessive alleles. You cannot be heterozygous dominant or heterozygous recessive. We'll talk about that in a little while, but that term does not exist. I hear it all the time. You can only, if you're a heterozygote, this will be the only thing you will see. This is homozygous dominant. This is homozygous recessive. Okay, but again, I'll talk about that when we really start doing Punnett squares in just a minute. So if the two alleles from an inherited pair are different, only one of those is going to determine the organism's appearance. That is the dominant allele. The one that does not have an effect on the organism's appearance is the recessive allele. Dominant are going to use uppercase letters and recessive are going to use lowercase letters. It's just the easiest way for us to denote them. And then the fourth hypothesis he came up with is that a sperm or egg will carry only one allele for each inherited character because the allele pairs are going to separate from each other during the production of gametes. You have seen that. Each of the 4 cells that are produced in meiosis all have half the DNA, so they can only have one allele for each character. We call that the law of segregation. They're going to segregate or separate from each other during gamete production, during meiosis. And then when the egg and sperm come together, the alleles will form pairs again, and that restores the diploid state. So a zygote, a baby fertilized egg, is going to be diploid. Now going back here for just a second with the homozygous, homozygote, and the heterozygotes. These guys, it's always kind of hard for me to explain. It's not like you're going to be homozygous for every single trait that you have, and you're not going to be heterozygous for every single trait that you have. So like you might be homozygous for eye color. If you have blue eyes, you have 2 of the recessive alleles. So you might be homozygous for that, but you might be heterozygous for your hair color or something like that, where you have 1 dominant allele and 1 recessive allele. So it's not like whole individuals are homozygous or heterozygous. It's more like the individual different characters, the different genes that you have, you'll be homozygous or heterozygous for them. Okay. And so here's a picture showing you the same experiment from the picture we looked at previously. It has a little bit more detail in here because it's kind of showing you the alleles segregating and what the gametes can give you. This is for you to look at if you need a little bit more detail on how he figured out the hypotheses. So I'm not going to spend a lot of time here, but if you look, it will tell you, like here, this comment right here is telling you how he figured out hypotheses 1 and 2. This statement is how he figured out hypothesis 4, and this one is how he figured out hypothesis 3. I'm not going to go over this, but if you want to look at it on your own, you can. So just some other terminology, the difference between a phenotype and a genotype. You may have heard of these things, maybe in middle school, and now you don't remember what they are. Phenotype is the organism's physical traits. The phenotype is what you see. So when I look at you, I see that you have brown hair, brown eyes, tan skin, you're 5'4", or whatever. That's your phenotype. That's what I'm seeing. That is what your physical traits are. The genotype is what the organism's genetic makeup is. So that would be like the letters, the stuff that we can't see. So in our examples that we've been using, the phenotypes would be white and purple. And then the genotypes are either 2 dominant alleles, so 2 uppercase, that's a purple plant. You could have a heterozygous, which is a capital and a lowercase, or a dominant and a recessive. That is also going to give you purple flowers because purple is dominant over white. And then 2 lowercase letters, 2 recessive alleles, those are the white flowers. And so this picture on this slide shows you both. So it's showing you the letters, but it's also showing you the colors. And then on the next screencast, I am going to show you how to do Punnett squares.